The next speaker is Christopher Large, and the title of his talk is Mechanisms of Hybrid Incompatibility Between Two Subspecies of Drosophila pseudoobscura.
Hello, everybody. I'm Chris Large, and I work in Nitin Fundis' lab at the University of Utah in the Department of Biology. Today, I'm going to talk to you. Move it up a little bit. All right. Is that better? Yeah? All right. And today, I'm going to talk to you about understanding the mechanism of hybrid incompatibility between two subspecies Drosophila pseudoobscura. All right, let's imagine an organism. Now let's imagine this organism is split into two different geographic populations that no longer mate with each other. Now they diverge over evolutionary time. However, they still have the same genes, the same development, and for all intents and purposes are the same organism. However, when you bring them back together, they now have incompatibilities that have evolved. This process of speciation is still enigmatic and is fundamental to both the fields of genetics and evolution. We studied this process using two subspecies of Drosophila pseudoobscura, one in western North America called Drosophila pseudoobscura USA, and the other down in Colombia called Drosophila pseudoobscura Bogota. They diverged fairly recently, less than 200,000 years ago. As a result, the only incompatibilities that have arisen are that the hybrid males are completely sterile and don't produce any functional sperm. However, within our system, when we age the males for 14 days, they become weakly fertile and produce only female offspring, a phenomenon known as segregation distortion. Previously, we've described the genetic architecture of this system, and we know that both segregation distortion and sterility rely on a complex genetic architecture, but that they both require a gene called overdrive for both the sterility and segregation distortion. This is important because there are very few instances where we know the genetic basis of speciation, and there are even fewer where we understand the actual molecular mechanism. However, our system is unique, as we do have overdrive as a point of entry to understanding the system. So we're going to approach this problem from two separate questions. The first is what goes wrong during male gametogenesis, and the second is what is the actual molecular function of overdrive. Now, before I get into the actual process of spermatogenesis within pseudoobscura, I want to orient everybody to the development. So it's very similar to melanogaster spermatogenesis, where you start with one stem cell that goes through a series of 5 mitotic divisions, and then goes through meiotic division, resulting in 128 spermatids. These spermatids then elongate and transition from a histone-based chromosome system to a protamine-based system, and then fully elongate and then individualize. So here's an actual pseudoobscura testis stained for DNA. It looks a little bit different from our picture, especially because there's way more information here. And because there's almost too much information when we just do a simple 3D projection, we decided to instead pseudo-color for depth so we can pull out each of the individual cysts that we're seeing. Because this is a high-resolution mosaic image, we can zoom in and look at each of the individual cysts and see what's going on. Here we can see the stem cells going to the mitotic divisions, going to the meiotic, resulting in the 128 spermatids that then elongate. I want you to focus in on this cyst right here and see that there is 128 spermatid there. This stage will become important later in my talk. All right, so returning to our original question, when does hybrid spermatogenesis fail? So we compare using stains for DNA once again for wild type and F1 hybrid. Originally, these appear like they're different. However, when we zoomed in, we've seen that they actually are completely the same, up to 128 spermatids. However, if we progress later, we can see that while the wild type has the beautifully elongated sperm nuclei, the F1 hybrid does not and has some sort of issue during condensation. Now, if we pull out these sperm nuclei, we once again stain for DNA, this time in cyan, and F-dactin now in magenta to be able to see the sperm tail. We can see the 128 nuclei all synchronized and compacted nicely near the front of the sperm. When we compare this to the F1 hybrid, we can see the nuclei are not compacted and are actually falling out along the tail of the sperm. So why do the nuclei look malformed? We decided to zoom in even closer. And what we saw was that the nuclei actually appear to have some form of chromatin condensation defect, that it looks almost as if there's a chromosomal structure, at least to me. So we want to figure out what is causing the condensed chromosome appearance of the nucleus. To do this, we decided to stain for the canonical condensed chromatin marker during mitosis and meiosis, phosphohistone H3 serine 10. This is labeled magenta for a wild-type pseudoobscura testis seen here, and then DNA once again in cyan. We can see a couple of the cysts that are going through synchronized cell divisions. Here's a mitosis up here, another mitosis, and then a meiosis. When we compare this to the F1 hybrid and remove the DNA channel, we can see that there is incredible more signal coming from the F1 hybrid. Initially, it looks like there's a lot of more overproliferation. However, when we've actually zoomed in, we've seen that it's exclusively within the 128 cell cysts. Essentially, what we're seeing is that the spermatids are failing to exit mitosis and remove this phosphohistone H3 serine 10. All right, so returning to our model of spermatogenesis, what I've told you so far is that the hybrid sperm nuclei have a chromatin condensation defect and a postmeiotic stall, and that this is associated with an aberrant histone modification. Next, I want to transition to talking about molecular function of overdrive, the gene that's responsible for all of this. So before I do that, I want to first talk about the domain structure. Overdrive has on one end a MADF DNA binding domain. On the other, it has a predicted protein-protein interaction domain. The way we wanted to uncover the molecular function of overdrive is to do CRISPR experiments making both native locus tags and CRISPR knockouts in both pseudoobscura and melanogaster. However, because melanogaster is such a great model organism, it just happened to work a lot better. And so today, the only data I have to talk to you about is melanogaster. So here we have our schematic for creating a CRISPR-mediated native locus gene fusion of overdrive with EGFP. The first tissue that we decided to look in was testes, as that's where we see our defect in pseudoobscura. We wanted to figure out where does overdrive localize during spermatogenesis. So this is a melanogaster testis. This time, DNA is in cyan and histones are in yellow. It's very similar to the pseudoobscura development, except that it's a spiral shape. So the stem cell niche is up here near the tip, and development continues around the length, ending near the center. Now, if we overlay overdrive and magenta, we can see that overdrive is a nuclear localized gene, but doesn't have any specific localization within the nucleus. It appears to be on from the earliest stages of spermatogenesis all the way through to the histone to protamine transition. So we want to figure it out, okay, is overdrive expressed in the germline stem cells? To this, we zoomed in on the testis tip and looked at the actual germline stem cells, and it appears that it is co-localizing with both DNA and histone 2AV, meaning that, yes, overdrive is present from the stem cells to the elongation stage. Okay, so we've seen that in pseudoobscura there is an effect during paternal germline, and that overdrive is localized to the paternal germline. So we wanted to figure out, okay, is overdrive doing something specifically in the paternal germline, or does it also have localization in maternal? So to do this, we pulled out melanogaster ovaries and stained once again for overdrive in magenta, DNA is in cyan, and now F-actin is in yellow. What we can see is that overdrive is enriched within the oocyte compared to both the nurse cells and the follicle cells. We can see this pattern holds even later into the stages, continuing all the way to one of the last stages where we see overdrive co-localizing with the DNA. So we do know that overdrive is localized indeed to the maternal germline. Next thing we wanted to look at is whether overdrive happens to, what happens to overdrive post-fertilization. So when we did this in live imaged overdrive here in magenta and with histones, histone 2AV RFP labeled here in cyan, we found that, okay, overdrive is maternally deposited and co-localizes with chromatin. But surprisingly what we found is that overdrive in fact disappears during metaphase and only returns upon completion of anaphase. So here I'm going to play a video of the development where overdrive disappears and then returns. Once more time, it disappears and then returns. All right, so returning to our previous data about the pseudoobscura hybrids, if we now overlay the melanogaster information, we see a pattern start to emerge. I'm going to come back to this at the end of the talk. All right, so the next thing that we wanted to do is look what happens when we actually remove overdrive and see if there's any issues during the development in embryos, ovaries, or the paternal germline. To this, we did a CRISPR-mediated knockout of overdrive and melanogaster. To our great dismay, however, when we looked at the fertility of the nulls compared to wild type, there was no effect. Neither was an effect on sex ratio on viability or non-disjunction. We found that actually overdrive is a completely non-essential gene. So now we have a list of incredibly disparate facts. We kind of got really dumbfounded here. We came up with the paradoxical statement that overdrive is not part of the developmental program, but can arrest cellular development. But this kind of starts to make sense if we come up with the conclusion that during gametogenesis, overdrive is acting in a quality control system during interphase of the cell cycle. If we then reconcile this conclusion with our pseudoobscura data, we come up with our model that overdrive is not being removed from chromatin in hybrids, triggering the control system, leading to a postmeiotic arrest. We're going to further test this using basically the same experiments within pseudoobscura and seeing whether overdrive does perdure within the hybrids. A statement I want to make real quick is that overdrive would not have been revealed in a simple genetic screen due to the lack of the obvious phenotype in melanogaster, and that only through studying an evolutionary question have we uncovered a potential important gene for the development of flies. And the last thing I want to leave you with, that through studying the speciation process during pseudoobscura, we've been able to reveal that chromatin dynamics play a critical role in the evolution of reproductive barriers between species. All right, I want to thank Dr. Newton Fudness for taking me in, and Nora Brown, who's going to be taking over this project while I head to graduate school at University of Washington, the rest of the lab, and Adam Lin from the Golick Lab, and then our funding sources, the University of Utah, Pew Charitable Trust, and the National Institutes of Health. Any questions?
Does it make sense to overexpress the overdrive in melanogaster?
Yeah, so we've actually done that, and we found that it has had no effect on pretty much anything, unfortunately, even at 29 degrees.
So are there different dynamics of overdrive returning differently between before NC13 and after NC13 in a transcription-dependent manner?
I'm sorry, I didn't hear that. Can you repeat a little bit louder?
Of course, at NC13 is when transcription starts. So before transcription, can you see overdrive return during interphase?
I'm sorry, I can't hear you.
All right. So before NC13, when transcription starts, can you observe the transcript coming back on in interphase?
In the embryos, is that what you're talking about? Can we see overdrive come back on?
During interphase prior to transcription initiation.
Oh, yes. Yeah, we can. It's maternally deposited. We just, histone 2AV doesn't actually come on until stage 5, so that's what we showed there.
Have you tried to overexpress it in the testes and see whether that has any effect?
We've done constitutive overexpression with actin and have seen no phenotype.
Have you seen differential effects on eusperm versus perisperm development in the hybrid crosses?
We actually haven't looked at perisperm.
Okay, thanks.
We have about a minute to kill, but it's okay. I did want to say that Chris is an undergraduate, and so when we picked him to give the talk, we thought it was quite interesting that we wish all our undergraduates would do as much work and generate these beautiful pictures. I'm sure that's not your experience with all of them. So I want to please give him. Thank you.